So today we officially start Unit 2, which is in AP Bio, cell structure and function. So in order to start this unit, we got to talk about the first very basic cells, and those cells are known as the prokaryotes. Most people know prokaryotes as bacteria. So bacteria is kind of like the common name for a prokaryote. And a lot of people think that bacteria makes you sick, which there are some species that do obviously induce illness, but there are also others that actually do serve some beneficial roles in our environment. Here are some common types of bacteria, prokaryotes, that you might have seen or heard about. Definitely Salmonella is a common one. We often find this bacteria on our food items, and when ingested, they can make you really very sick. Streptococcus is what causes strep throat. So if you've ever been sick with strep throat, that's the bacteria that was infecting you. Of course, we have our E. coli up here in the top right. And so all of these are different types of prokaryotes that you might have seen or heard of recently. Before we dive into what makes up a prokaryote, let's look at some of the history first. There were many people who were working on developing the microscope, but the man who was pretty much given credit for taking and modifying the invention to become the microscope, his name is Anton van Leeuwenhoek, and he kind of invented, if you want to call it that, in the 17th century, which was about the 1600s. So once you had the microscope, then you could see things and magnify them to see smaller things that normal people couldn't see with their eyes. Here is a picture of a Leeuwenhoek microscope. So you can see it is quite small. And then here is an image that shows you what it looks like in comparison to a human face. So you can see it was quite small. You held it up to your eye and then you put the object behind it so that you could magnify what was being looked at. Now, where did the name cell come from? That was given credit to a guy named Robert Hooke. He was using one of those microscopes and he was looking at dead cork underneath the microscope, and he's like, oh, those kind of resemble cells. Now, there is some story to why it was named a cell, but I think most people will agree that it is because it looked like monastery cells, so places where people slept in a monastery. Once cells were seen and named through the use of the microscope, we got the creation of what is called cell theory. And basically, cell theory is composed of three parts. The first thing says that all living things are composed of cells. So any sort of living item has cells in it. Two, cells are the basic units of structure and function in living things. So they're living and they are working and doing things to make this living function work. And then the third thing is that all cells come from pre-existing cells. So you wouldn't have new cells if it wasn't for the older cells dividing, creating these new cells. There are different types of cells, as you're going to learn about through this unit. You're also going to see that the cells have very specific structures, and that will lead to the function of that cell. One of the first types of cells that we're going to talk about are the prokaryotes, and these are the cells that are classified as the bacteria. You also have your eukaryotes. Those are different types of cells. They're more complex, and we'll get into those in a little bit later. But they are primarily the plants and the animals and the fungi. And then we have this not so cell structured thing called a virus. And a virus is considered not living because in order for it to replicate and divide, it needs a living host. So we don't really classify it as a type of cell, but it does exist and it can be found in living cells. So just a little bit more about viruses since we are on the topic. So viruses aren't considered living, and again, that is because they do need a host in order to survive. So in order for viruses to live, they have to inject their genetic material into a host cell. That genetic material will instruct the host cell to make more viruses, and then they replicate that way. What you're looking at is a very cool looking virus. It's called a bacteriophage virus. And what this virus does is, even though it kind of looks like a spaceship, it does actually infect bacteria. That is the main host cell of this virus. All right, on to the prokaryotes. So prokaryotes are the earliest cells, or thought to be the earliest cells here on Earth. They are extremely small when you're comparing them to a eukaryotic cell, and they don't have a nucleus. That's kind of like one of the main characteristics that make it a prokaryote. So it doesn't have a nucleus. It doesn't have this nice little structure for its genetic material. But it does have some other things that we'll talk about that make it distinctly prokaryotic. So one item that all prokaryotes have are cell walls. And the main function of a cell wall is pretty much for protection. So you can find a prokaryotic cell wall on the outside of the cell, and it's usually made of a chemical called peptidoglycan. So you got peptide, which means it's protein, and glycan, which means there's some carbs. So using our knowledge of macromolecules that we just acquired in Unit 1, you can see that this is a combination of both of those macromolecules. Right underneath the cell wall of a prokaryote, you will find what's called the plasma or the cell membrane. And the plasma of the cell membrane has a function. It's not necessarily to protect like the cell wall, but it's more like a regulator. So it will allow things to move in and out of the cell. We'll learn more about what can come in and out of the cell and how this works in a couple lectures from now. Just know that the cell membrane is considered semi-permeable, so that means some things are let in, but not all things. It's made of a lipid, so tying into Unit 1 are macromolecules, and it's a bilayer, so that means it's composed of two layers, as you can see here in this image. So prokaryotes also have genetic material, which is deoxyribonucleic acid. Again, tying into Unit 1, our fourth macromolecule, the nucleic acids. So like I told you in the beginning, prokaryotes, they don't have a nucleus to contain their genetic material. However, wherever you find the genetic material in a prokaryote, it's usually termed the nucleoid region. So it's not really a structure per se like you see in the nucleus, but it is a region where you can find the genetic material. The genetic material of a prokaryote tends to be small and it tends to be circular. And then prokaryotes also tend to have these pieces of extra DNA that are also kind of circular in shape, and they're called plasmids. And I'm introducing this now because as we get later on into our units, you're going to notice that a lot of bacterial manipulation and engineering occurs with these plasmids that are found in prokaryotes. Prokaryotes also have ribosomes, and ribosomes are usually free-floating in the cytoplasm, which is the fluid of the cell. And ribosomes obviously are super important because they are the site of protein synthesis. So it is where proteins are made. A lot of prokaryotes will also contain items that help them move. And so one example of that is the flagellum or flagella for plural. So flagellum tend to help propel the bacteria in areas and pretty much just help them move. So the question arises then, how did eukaryotes develop if they're more complex and we still have these prokaryotes around? Well, The answer lies in what's called the endosymbiotic theory or endosymbiosis. Both are correct terms. And so the theory pretty much states that there were these different types of bacteria. Some were larger than others, and a large bacterium, a prokaryote, engulfed another smaller one. And instead of destroying that bacteria, it simply lived inside the larger bacteria. So it engulfed it and they were symbiotic with one another. And so as that bacteria, the engulfing bacteria, divided, it also passed on the thing that it engulfed. And so from that, you got more complex cells that we now called the eukaryotes. So in essence, prokaryotes pretty much paved the way for the more complex eukaryotes. So remember, prokaryotes, they're small, and then when you had those get put together, they made these larger, more complex cells called eukaryotes. And of course, we will be talking about those much later. So I hope all this was good. Remember to take your notes and submit them to Schoology.